For everyone with an interest in NASH, or more broadly, fatty liver disease, Surf's Up, Season 3, Episode 41 of Surfing the NASH Tsunami. Our look at practical challenges in clinical trial strategy and design today starts now.
This week on Surfing the NASH Tsunami.
We used to do a lot of biopsies, and in clinical practice, looked at the biopsy, then pulled out the biopsy again, cut slices off that piece, and sent it off for central review. These days, we're much more focused on actually NITs. Even in most cases, I don't have a baseline biopsy to consider the patient eligible.
And I've seen that. For instance, we have an ongoing obesity trial. It's not a NASH trial. It's an obesity trial with the same drug we're using in one of our NASH trials. The screen fail rate's only 25%, as opposed to a NASH trial that has screen fail rates of 80%. And you can see that the energy around enrolling that obesity trial is so much higher.
Stephen hit the nail on the head when he talked about sites getting disheartened when you don't get people into studies.
You could be in a downtown urban environment and still not have access to NASH clinical trials. Think about Chicago. If you look at the number of NASH patients that are enrolled in clinical trials that come out of Chicago, you would think that that city is the thinnest, healthiest, most athletic on the planet with no genetic predisposition to fatty liver, because we're putting almost zero NASH patients in clinical trials.
In behavioral psychology, the single pattern that is least likely to produce positive behavior is a low and inconsistent success rate. So if I harken back to Stephen's comment about the obesity trial, the simple fact that you can drop a screen fail rate from 80 to 25 means that the people doing the recruiting are going to get a lot more frequent positive stimulus for what they're doing.
You would think that sticking a harpoon in the side of somebody and telling them that they have disease or that they don't have disease would be a real motivator. Unfortunately, I'm not sure I've seen that pan out in clinical practice.
If you think about medical care, there's a big difference between Europe and the US. You might say, the things that Stephen are describing is a lot based on inequality between groups, but it's not. It's the same fact, and even in socialized healthcare like Germany, that these patients are not accessing NASH trials because the hurdles are at multiple levels.
If you want to change your rules, find more juice. The question then becomes, is the juice worth the squeeze? The two specific forms of juice is going to take care of cash and optimism. Optimism is going to motivate people to step out of the boundaries they know, and cash because it's going to give them the tools to do that. And what will make the juice worth the squeeze is one particular drug succeeding.
Every week, a global community of fatty liver disease stakeholders comes together to explore the most important challenges in diagnosing, treating, and developing medications for patients with fatty liver diseases. Join the Bedrock Surfing NASH crew, hepatology researchers, and key opinion leaders Dr. Stephen Harrison and Professor Jörn Schattenberg, liver wellness advocate Louise Campbell, and pricing and forecasting guru Roger Green, as they discuss new approaches to practical challenges in clinical trial strategy and design on Surfing the NASH Tsunami.
So this is a fantastic episode. I don't remember the last time we had an episode that was just Stephen and Jörn and Louise and me. I think we did one of those a little bit ago. And then before that, you'd have to go back to like the middle of last year or something to find that particular combination. But here we are all today. Jörn is back from vacation. Welcome back.
Thanks, Roger. Glad to be here again. 2 weeks of being off of clinic. It's a joyful moment to join you guys again here to record for the podcast.
And we're thrilled to have you with us. Going clockwise on my computer, I guess I get Louise next. Louise is now working on her second computer of the day to get a podcast, but she's here. How are you?
I'm very well, thank you very much. Nice to be with everybody again, and nice to see Jörn fully refreshed after his holiday, and Stephen, I haven't seen for a while. Nice to have you all back.
So Louise has two things she's looking forward to. She has Liverpool Crystal Palace, which will kick off in about an hour and 45 minutes, her favorite team and her second favorite team, or her husband's second favorite team and first favorite team, depending upon which half of the couple you're looking from. And then next week, she's going to be driving the entire boat on this podcast, moderating, working with our new associate producer, Jake Norman, to get all our print materials and stuff like that together. And we're going to see how that goes. We're going to take a run at it and see what happens. And then finally, we get to Stephen. Hey, Stephen, how are you today?
Hey, Roger. Hey, Louise and Jörn. Great to be back. Great to be a part of today. Just suffering through a long, hot summer in Texas.
Yeah, I understand that. I was sharing with Stephen the data point I'd read recently, which is that San Antonio averaged 5 days over 100 degrees Fahrenheit, 38 Celsius for those of you in Europe, until this year. And this year, there have been 55 such days. So that's a lot. And then as far as I go, I'm good. I'm here back at the Jersey Shore with family and now and friends later, and just actually enjoying the balmy breezes in the ocean. With that, why don't we go to Groundbreaker? We got one good thing, personal or professional, that happened to each of you in the last week. Brave one, go first.
Oh, well, good for me, although a bit intimidating, as I've been asked to write a NATHLed chapter for a book that's going to be published for nurses. So I've never written really academically other than my dissertation and things like that. And I've certainly never written a chapter for a book. So this is going to be interesting. It's quite a short timeline, but I may well be calling a few of the top gurus that I get to speak to just to give me the insight. That's my professional, and my personal is that we've booked our next course for sailing, so hopefully I'll be a day skipper at one stage, but that's a long course and a lot of work.
I figure about the time I next get to England, you'll be able to take me out on a boat. That'd be fantastic.
Well, I'll be taking you on a Viking ship.
Ooh. That's fantastic. Okay.
Yeah, I'll give you a quick refresh from my last 2 weeks, because we spent time with family in Italy. Northern Italy. It was kind of hot too, but it was too hot for me to go running. So my personal highlight just before I joined you guys here, I actually took a 10K around the fields or the vineyards here in Rhineland-Palatinate. And so I'm very refreshed and energized to kick off.
Jörn, what's the name of the drink that's 1 part mineral water or sparkling water and 1 part white wine?
Well, so they call this a Schorle here, Schorle. So depending on where you are, it's more white wine than sparkling water. It's very refreshing, of course, and something that's typical of this area.
I just figure, given what time of the day it is there and that you've been through the vineyards for 10K, I know where you're headed when we get off the podcast, which should be just fine. Stephen, you're next.
You know, it's that time of the year where school starts back, and my 2 college kids are headed back to their respective colleges, and we're getting them all set up for the fall semester. And we just took our daughter back to Southern Methodist University in Dallas, where she is on the cheerleading team there. So we were getting all that situated and ready to go. And soon we'll be empty nesters again, 1 more week, and it'll be me and my wife and 2 dogs. So looking forward to that time as well. But glad we had the kids home for the summer, and now we're glad to see them back in school.
Perfect combination, isn't it? Going both ways. So on my end, I'm starting vacation down at the shore, which is my favorite time of year, except when I get to go to Park City and ski and hang out with all you guys and do NASH Tech, which I also find a fantastic time. And we rolled out the CME podcast last week. Episode 2 of the Diabetes podcast is coming out this week, and life is good. So we're going to get started. We announced a couple of weeks ago that what we were going to do for this episode was we were going to focus on going back to Jörn's paper from 2020 about the NASH drug graveyard. And some of the people that we had wanted to have join that episode were not able to make it. Fortuitously, Stephen was. So we're going to do about half of that now, and we'll do half of that sometime in middle or late September. The half we're going to do now is this episode is going to wind up taking a look at issues that reflect on cost efficiency or cost effectiveness of getting NASH clinical trials done, particularly looking at 2 different issues. First of all, issues around patients, patient demand versus patient supply. And then if you can get enough patients into a study, screen fail rates and what they do to timelines, which we'll come back to again in a minute, and what they do to cost. And then the second issue is length of process. How long does it take to get a drug through trial, given that the longer it takes, the more money the manufacturer is spending and the less revenue they are realizing. So we've got 2 of the best to talk about those issues today. And that's what we're going to do. We're going to start by having Jörn talk about some patient issues, and then Stephen talking about some other patient issues, and we'll trial process. I'll have questions, and she'll have questions. And with that, why don't we take off? Jörn, welcome back. And after the run and before the Charles, is that how you pronounce it? You'll have to.
That's pretty close.
Okay. Not bad for a kid from New York, and who speaks no German, and somewhere between the run and you have time to do this. So why don't you get us started?
Thanks, Roger, and I'm happy to have Stephen on here because. The time I oversee in drug development in NASH is significantly shorter. Still, I've seen my share of trials fail or being terminated early, so that's of course very painful. And if you think back a number of years, in the paper you're referring to was one I co-authored with Joost Drent in December of 2020. We discussed why some of the drug programs failed, and that was fresh under the impression of even large phase 3 trials being halted. I think we've learned a lot since then, and clearly clinical trials in NASH today are very different. The discussions are driven by different issues, and some of them we've discussed at length in the Hot podcast here. I think the way we talk about endpoints and biopsies is very much different. The way we use biomarkers and artificial intelligence to augment outcomes has changed the field dramatically. But in the end, we still are linked to an endpoint that's a surrogate for the regulators that's required for conditional drug approval in this arena, and all phase 3 trials are actually looking at that endpoint. So in the end, it's not that much different. But what's different is I think rethink the way we select patients and the way we talk to patients about the trials. And that might be something to revisit and actually get different perspectives, particularly Stephen's, of course. And I can give you my perspective on how we select a patient today for a trial. Which patient for which trial is always an interesting aspect. If I go, if I have 2 or 3 trials running and patients eligible for both, of course, pre-screening activities, considerations, predicting the baseline biopsies, all these things. And I think that has changed significantly compared to the earlier trials where we just went for the biopsy and looked at what we had. So I think it informs the drug development process and will also eventually lead to a successful ending here for one of the phase 3 trials we're running right now.
Jörn, could you go into a little more detail maybe on what you think are 1 or 2 of the most important things that have changed or you've improved or figured out as a result of all that and the various conversations everyone's been having?
Yeah. So from my practice, and I think it's a little different in clinical trials from my practice, I used to rely heavily on historic biopsies to decide if a patient is eligible for a clinical trial. I think this is a very valid approach. We used to do a lot of biopsies and in clinical practice, looked at the biopsy, then pulled out the biopsy again, cut slices off that piece and send it off for central reviewing. These days, we're much more focused on actually NITs. Even in most cases, I don't have a baseline biopsy to consider the patient eligible. And we're using NITs as the diagnostic biomarkers to predict both the presence of steatohepatitis, ballooning, and a certain degree of fibrosis to then biopsy a patient. That leads to a situation where you have more liver tissue going into the study, which I think is very crucial because you have a better assessment, stronger assessment of the baseline biopsy, not a short or fragmented piece of biopsy. And it also enriches for at-risk patients because clearly depending on which NIT has been considered, it brings in a certain dynamic for the patient population that's then enrolled in clinical trials. So I think this is my first thought that has changed for my center over years, and maybe if somebody from the team wants to reflect on how they do that. I know some investigators are still heavily relying on historic biopsies, but clearly for me that has changed over the years.
So Stephen, as Jörn pointed out correctly, you see a lot more of this stuff from a lot more places than Jörn as a prodigious but single-site investigator does. To be fair, reflect a little bit on his comments and explain to everybody what you're seeing that has changed. If you've got specific things that some people are doing that you think are working well that could be more broadly disseminated, this would be a great place to share that. Your floor.
Yeah, I think the comments that Jörn made are kind of where we are today as a collective group. We historically did a lot of liver biopsies. I remember the day I did 5 a day, 25 or 30 a week. We would wait for the results to come back. If they had bad NASH, we would entertain screening them for a clinical trial. And as Jörn said, we would carve off a couple unstained slides off the block and send them in and wait for the result. And I think either because we clearly don't do as many biopsies anymore because we rely heavily on non-invasive testing. In my clinical practice, I use FibroScan and liver chemistry tests. I use the FAST score. I order CT1 and MRE in many of my patients. And more, we're using these specific cut points to decide who we refer to clinical trials. So not just in my practice, but I think in many practices, this is the way it's done. So if I see a patient and they have an elevated AST, they have an abnormal FibroScan, often I'll order a multi-parametric MRI. If the value comes back at 900, 950, something like that, above 875, that person almost assuredly has NASH with some degree of fibrosis. If it correlates with the FibroScan, then I'm going to be referring that patient to a clinical trial, at which point the patient would then be screened in, undergo the requisite labs, as well as MRI, if that's part of the study, and then onward to liver biopsy, where we're still kind of relegated to a yes-no decision by a central pathologist. And we'll talk more about that, but I think that has been another point of change over the past year or two, for sure. One of the ways that corporately we've seen a change in clinical trials relative to this non-invasive testing strategy are some very specific cut points that we've been able to use in real time to help redefine for clinicians what's working relative to the pathologists that we're working with in a clinical trial. And let me give you an example. So let's say we start off a clinical trial. It's a phase 3 trial. We need to put 1,000 patients in it for Subpart H approval, and we have the inclusion criteria set. We all know that's a NASH patient with an NAS of 4 more with F2 or greater fibrosis. And we start off by saying, okay, we know historically that there are going to be very, very few biopsies already done. We mentioned that already on this podcast. So most people are coming in based off a pre-screen strategy. We used to refine that and define that as an AST greater than 20 and a KPA on FibroScan greater than 8.5. Patients that met those criteria were sent in, biopsies were subsequently done, and they were either screened in or screen failed. Now, what we can do is refine that a bit more. We have something that we readily use in clinical practice called the Myfibroscan App. It's on our phones, and we can simply put in the AST and the KPA and the CAP score and generate the FAST score. And we use that to help guide whether or not they're going to meet criteria for a biopsy. And along the way, we can mine that data in real time to say, okay, these are all the screen fails on liver biopsy. These are all the screen successes on liver biopsy. Let's look at the FAST score for those that made it into the trial. And so we do that at intervals now along the way, where we're constantly adjusting fire, if you will. We're resetting true north to say, okay, what FAST score seems to be working? For some, it's 0.51. For some, it might be 0.54 or 0.56. But we know that adjusting that in real time gives us a higher probability of success on biopsy. So over the years, and I would say particularly over the past year, we've been able to take a baseline NIT and refine it midstream to meet what our pathologists are requesting for inclusion on clinical trials. That's been really, really helpful. And we'll talk about the pathologist piece in a bit, but I wanted to just kind of address Jörn's comment on NITs and how we're using them in clinical practice, how we're using them in clinical trials, and how really they have become very helpful, even in the middle of a clinical trial, to adjust fire a little bit to meet the nuances of what a particular pathologist is looking for when they look at these slides under the microscope.
Stephen made some important comments, and just before I hand over to you guys again, maybe I follow up and say the NITs we're using are not always necessarily part of the protocol defining inclusion or exclusion. It's kind of the state-of-the-art how an experienced NASH center would evaluate a patient and consider him for enrollment in a clinical trial. Now, we've seen some studies, in particular phase 3, that are pre-selecting patients based on metabolic risk factors, some very clinical aspects, obesity, presence of diabetes, not so heavily coined towards NITs, and that, of course, reflecting that it's not always available everywhere. In particular, if you want to enroll a large amount of patients in a relatively short time, you might not be able to go to those very specialized centers. On the other hand, I think it helps tremendously to refine it. And for a sponsor, I think even if you do not require a certain NIT cutoff, it's important to capture those, because the investigators, in my experience, are using them to direct a patient towards the trial, and you're going to have a certain pre-selection. If it's in your protocol or not, it's going to happen.
And that's a good point. For sure, these NITs are not necessarily always built into the inclusion and exclusion criteria for a study. They're both. Sometimes there are specific metabolic risk factors plus NITs. Sometimes there are just metabolic risk factors. But the whole idea behind this is we clearly have learned that we don't just want to take any patient with fatty liver and screen them for a trial. We know that if we enrich them for diabetes, obesity, hypertension, even evidence of early renal dysfunction, that we're very likely to have an enriched population that would qualify on liver biopsy. And that's a good thing, right? We don't want to do unnecessary liver biopsies. And so if we can continue to refine the right patient to bring forward for a study, that does several things. It obviously keeps the site engaged. They don't get what we call study fatigue, where they have so many screen fails, they've been burned out. The coordinating team stays motivated. Patients are more apt to be engaged. And I've seen that. For instance, we have an ongoing obesity trial. It's not a NASH trial, it's an obesity trial with the same drug we're using in one of our NASH trials. The screen fail rate's only 25%, as opposed to a NASH trial that has screen fail rates of 80%. And you can see that the energy around enrolling that obesity trial is so much higher than the NASH trial, where where we're struggling to find the right patients to cross the finish line. So of all the things that we have honed in on this podcast and in real life and at liver meetings such as EASL and AASLD, probably the most important one, in my opinion, is finding ways to mitigate that screen fail rate and finding the right patients to qualify. And I'll just say it now because I think it's apropos for this part in the discussion, is where we've learned this challenge existed dates back a couple of years, but clearly we know that there's heterogeneity in liver biopsy, there's sampling variability, there's interpretation variability amongst pathologists, and we lost a couple drugs, whether it's the MSDC0602K drug, that's the one that stands out, or seladelpar, which is another one that comes to mind. Those drugs were lost in part because they were read by a single pathologist, and unfortunately the methodology brought forth in both of those contributed to the demise of those drugs in the setting of NASH. For MSDC0602K, it was rereading the baseline biopsy to try to eliminate temporal bias, and in so doing, a quarter of the patients on reread by the same pathologist were found to not have NASH at baseline, when earlier the same pathologist said there was NASH at baseline. So when you imputed the statistics from that point, the drug did not have statistical significance for NASH resolution, despite the fact that there was a dose-response relationship. When you actually went back and looked at it the old way, there was statistical significance. That drug would be in phase 3 today. With seladelpar, which is a very similar situation, single pathologist read of the baseline. At the time that the end of treatment biopsies were brought forth to read, a second pathologist was brought in to help. That second pathologist found some issues in the follow-up liver biopsy that were concerning in the pathologist's mind for drug-induced liver injury. That prompted a halt to the trial and a big investigation where all the baseline biopsies were reread, and it turns out that those same lesions were there already on the pre-read as on the end of treatment biopsy, and had that been adjudicated at the beginning, that drug would have continued on and very likely would be in phase 3 for NASH now. Well, just to continue the thought and fast forward, we've learned that potentially it's better if more than one set of eyes looks at a biopsy, and so we've begun down the road of consensus read, and we're not fully vetted yet on what the best way to do this is. There are some studies where you have two pathologists, they both read independently. If they agree, great. If they don't, they come together and adjudicate the biopsy collectively and get what we call a consensus read. There are others that have three pathologists, each of them read independently, where two agree, where none agree, they get together on a phone call. And then you have an additional situation where AI digital pathology is being done and being provided to the pathologist as what we call a companion diagnostic, so they can look at the computer-generated report as they adjudicate their ordinal reading on the liver pathology tissue. And that's kind of where we are today, is some variants of that last little bit. Two pathologists adjudicating consensus, three pathologists adjudicating consensus, or a situation where in either of those scenarios there's AI digital path being brought to the table as well. What we're not, and I want to make sure this is clear, where we are not yet is fully AI digital path read. In other words, a computer replacing a pathologist. That has not happened and probably won't happen. I think where we're headed in that is we'll transition to non-invasive tests, but use AI digital pathology to help us gain a better idea of ground truth as we transition to a non-invasive testing strategy. But I think that's another area where we've come significantly and made huge strides, maybe in the past year even, I think.
I wrote down 3 notes, but Louise, you have any questions you want to share before I dive into those?
No, I think everything that the guys have been saying is absolutely spot on. From a different side, Stephen hit the nail on the head when he talked about sites getting disheartened when you don't get people into studies. Stephen runs a big site, but we were on sites, and I know other people at sites, where you only have 5 slots. If you don't recruit to these slots, and it's very difficult, the first people that the patient engages engages with are nursing staff. If they're discouraged by the way the trial is running or the lack of ability to recruit, then that message gets across, because it's very difficult to try and encourage somebody into studies. We need to keep that interest up and that vigour, I suppose, for the patient recruitment. And it's just absolutely vital. I listened to your presentation at ILC, Stephen, where you looked at raising the threshold for Fibroscan, for example. I think it was 12.9 with a higher cutoff for CAP as well, in that context that by the time you remove and you drop the AST down, then you will also see the fibrosis drop. So I was interested in that, and I think that's what I would certainly see traditionally in patients. The higher the Fibroscan, by the time you account for the AST as it normalizes, then you see a more true picture on biopsy. That was actually quite important, because I think the cutoffs now are still, as you both described, 8 to 8.5, which suggests you might get a high screen failure. But I thought that was an important session that you did at ILC that explained that.
I don't think we have a cookie cutter approach to this. I think each clinical trial is different. It has its own ecosystem, if you will, of what works or what doesn't work. It's contingent on the pathologist that's reading the slides. It's contingent on the criteria that you have to meet to enroll the patient. But I think on a very standard phase 2B or 3 trial where you're looking for NASH patients with an NAS of 4 or more and the standard F2 to F3 fibrosis, we are able to get a little closer to where we need to be using our non-invasives. I see a role potentially for NIS4 here as well. That's kind of designed to find the F2-3 patient with an NAS of 4 or more. I see a role for Myfib, which is Rohit Lumba's criteria. I see a role for MAST here, which Mazen Noureddin brought forward. Those last two involve MRE. So if you're not getting MRE as a clinical tool or MRI PDFF, then you're not going to be able to use Myfib and MAST in your pre-screen strategy. But NIS4 is a blood test that could be sent off. ELF is another one that one could look at to help stratify patients. Of course, ELF doesn't tell us anything about NASH. It just tells us about fibrosis. But you don't have to just use FAST like I commented on. There are others out there that could be done, and we could develop a strategy in real time to help guide the cutoffs. For instance, if you're using NIS4 and we're a third of the way through the trial and we can analyze screen fail data on biopsy versus screened in data, we can come back and analyze what the optimal cutoff is for NIS4 to meet eligibility for this particular trial using these particular pathologists. That could be done in any non-invasive, not just FAST, not just NIS4, but Myfib, MAST, anything where we are able to obtain clinical data to be used as a pre-screen strategy for enrollment in a clinical trial. So I do think the more we can do that, the better off we'll be. But that gets us to another issue that I think we wanted to talk about on this podcast, and that is finding these patients. If you think about all the NASH trials that we're doing and all the NASH trials that we've done and the thousands and thousands of patients that we've gone through, there's only so many people that are going into the NASH clinical trial funnel. It doesn't mean there aren't millions of NASH patients out there. It's just they're not able to access that funnel. They're in rural areas, small towns. They don't have transportation. And so maybe they see a primary care doc who doesn't know anything about research, or a GI doc that doesn't know anything about research, or they don't live in an area that's adjacent to a brick and mortar research facility. So you could be in a downtown urban environment and still not have access to NASH clinical trials. And I guess I'll just say it here on the podcast. I'll just give you an example. Think about Chicago. Chicago is a huge city, one of the top 5 cities in the United States as far as size goes. If you look at the number of NASH patients that are enrolled in clinical trials that come out of Chicago, you would think that that city is the thinnest, healthiest, most athletic on the planet with no genetic predisposition to fatty liver, because we're putting almost 0 NASH patients in clinical trials out of a city as large as Chicago that has high rates of obesity, high rates of diabetes, high rates of metabolic syndrome. And so it's not just a rural environment. We could drive across the United States and go through small town after small town where there's no access to NASH clinical trials, and you could say that's that's the reason. But it's not. It's a much bigger issue than that. And so circling back to my comment on the NITs, these people, even if you were able to access them, they're not getting FibroScans. They're not getting MRI PDFFs and MR elastographies. And the providers aren't educated in NASH diagnostic tests to know to order an ELF or a NIS4 or to calculate a FAST score or to use a Myfibroscan app. So we're kind of relegated to the tried and true, getting after the brick and mortar centers that we do know about to teach them how to locate these patients, how to pre-screen for them. But I would argue that we were missing a big opportunity in targeting people that heretofore have never been offered an opportunity to do a clinical trial. So what do we as a community need to do to reach out to those patients? And how do we do that? And who do we need involved with that? Sponsors, CROs, regulatory authorities, patient advocacy groups, the podcast, everybody together? I don't know, but that's a huge area of opportunity for the patients as well as for the sponsors and as well as for the drug development community as a whole that we haven't tapped yet. We're just kind of going back to the same old well over and over again, and that's delaying our ability to enroll these trials.
You know, if you think about medical care, there's a big difference between Europe and the US. You might say, you know, the things that Stephen are describing is a lot based on inequality between groups, but it's not. It's the same fact, and even in socialized healthcare like Germany, that these patients are not accessing NASH trials because the hurdles are at multiple levels. So clearly, even coming from a socialized healthcare system, I do see the same gatekeepers or the same problems for patients. They're not reaching the physicians offering those treatments.
First of all, Jörn, in some ways, I would probably argue that for all the inequality in the U.S., there are things about the U.S. system that make it easier to get it right here than there. But I'll come back to that in a sec. My first thought as I was listening to Stephen, I have 3 or 4, but the first one is, you know, in behavioral psychology, the single pattern that is least likely to produce positive behavior is a low and inconsistent success rate. So if I harken back to Stephen's comment about the obesity trial, the simple fact that you can drop a screen fail rate from 80 to 25 means that the people doing the recruiting are going to get a lot more frequent positive stimulus for what they're doing because more patients are going to get in, which goes back to Louise's comment about the nurses. What that says is the simple act of dropping more patients in the top of the funnel doesn't solve the problem, because if you drop more patients in the funnel and you still have hit rates that are way too low and where people aren't very good at understanding where they're going to hit and where they're not going to hit, then all you're going to do is you're going to reinforce patterns of failure, and it makes it harder in and of itself for that to succeed. It's a vital thing that we get more patients poured in the top of the funnel. It's equally or more important that we become more systematic and successful about getting patients once they're in the funnel through the funnel. So if I pull that back to the comment about the US versus Europe, the more technology we have and the easier time we have distributing cutting edge information within the medical system, the better job we're going to do staying on top of the later advances. And that would tell you that the US might do better because we spend more money on it here and we've got more technology. So as you all learn the things you all are learning, the Americans have a better shot at putting it to play faster than the non-Americans do, because frankly, just because of things like you got more MREs available in the States than you do on a per capita basis than you do in other parts of the world. So that would be thought number 1. Thought number 2, and this is a question, is the focus of a lot of our conversations on these topics over the last couple of years has really come down to screen fail rates, what improves them and what makes them worse. And today, Stephen raised a concept that's never been on this podcast before, which is mid-trial adjustment, kind of moving your fire mid-trial based on what you're seeing. 2 questions. First of all, are we having success getting screen fail rates down with all the steps we're talking about? And then second, I'm going to want you to come back and talk about what that means, because it's just not something we've talked about here at all. So screen fail rates first and then mid-fire second.
I think it has helped with screen fail rates some. I think it's trial specific because in a way it gets back to a comment I made numerous times in this podcast, and that is, how many psychiatrists does it take to change a light bulb? And the answer is 1, but the light bulb has to want to change. So we can provide sites study data in real time and have a principal investigator, a coordinator call where we disseminate that information and say, look, if you make this change today, in your screening practices, you will have a better success on liver biopsy. And there are sites that take that to heart and do it, and there are other sites that just keep doing the same old way. But where they enact that, where they institute that change, there is differences that are being seen. So just maybe a little bit more about what that means, Roger. So if we start out and we say, okay, let's screen patients that have ASTs greater than 20 and FibroScan KPA is greater than 8.5 and CAP score is greater than 280 to identify the NASH patient with F2, F3 fibrosis. We do that for maybe 200 or 300 screens, and we get 150 liver biopsies, and 2/3 of them are screen fails and 1/3 of them screen in. What we're doing now is we're taking all of that data and we're applying it to our non-invasive tests that are readily available to the people that are in the clinical trial. And so I mentioned earlier the CAP, the FibroScan, and the AST. Well, so there we use the FAST score because we know that has been designed to find that same patient, a NASH patient with F2, F3 fibrosis. And the ideal cutoff is 0.67 in the clinical trial that we published 2 years ago now. Well, as an example, when we apply the data in real time, what we see is that, yes, you could use a cutoff of greater than 0.67, and you have a high likelihood of getting patients in the trial. The problem is you're going to leave a ton of people that would otherwise qualify for the trial behind. So when we look at that data in the context of what I just mentioned, what we find is that there's a lower threshold for FAST score where we still get a high sensitivity and specificity, or an ideal Uden's index. And for a trial we just did, for instance, it's 0.51. So it's not 0.67, it's 0.51. That gives us a better shot on goal of capturing more patients and qualifying them with an 80% chance of a success rather than a 70 or 80% chance of failure. But there is a sweet spot on the scale. You don't want to have your score so high that you enroll everybody that you bring in, because then you're not going to screen a lot of people, and many people that would otherwise have qualified are being left on the sidelines. Vice versa, you don't want to set your criteria so low that you screen everybody and your screen fail a ton of people, they go to biopsy unnecessarily. So finding that sweet spot has been the challenge, and that's why we do this real-time exercise a couple times in the middle of a study to show, do we need to go up or do we need to go down to maintain an optimal screen fail rate, at the same time making sure that we're not letting too many people in and we're not bringing in enough people.
That's fascinating. Have you all taken a look at whether cohorts brought in at one level or another in the same trial have different performance within the trial?
Now, we haven't, as far as results in the study, we haven't taken the data out that far yet.
Didn't feel like you've gotten to the finish line. That it seems to me like that's the piece that seals that deal, right? If it turns out that as you make those adjustments, it doesn't have a lot to do with how well the drug performs.
Right.
Then you don't have to ask a question of what the right answer is. The answer is it's all the right answer. Well, that's fantastic. Thank you. Let me go back to everyone else. Last questions before we go on to the other topic we wanted to spend a little time on today.
Well, I think there's a little bit of the risk that we're overfitting our patients. On the other hand, we want to really, as Stephen said, sweet spot the amount of patients we want to still enroll because they have a liver disease that might be amendable or druggable. And it's a tough decision to be made at the beginning. And as you rightfully said, it'd be very interesting to actually look at, maybe that's a predefined analysis you could include in a clinical trial, look at how the NITs perform in terms of enrollment and then adapt and go forward and agree on that with the regulators from the start. So that's a nice thought that should be taken forward.
I think there's another part as well when we talk about the recruitment. Often sites will use we're going to give you 3 to 6 months to turn around and see if you can change your lifestyle and we see an improvement in your FibroScan, your other NITs. If that happens, we don't progress to biopsy. There is a way of looking after the patient that also can remove people from a screening timeline and into biopsies. And I wonder if anybody's ever analysed the effect of the liver biopsy on the screen failures as to how that motivates lifestyle change. Because most people do not want a biopsy and will do anything to avoid it. So where they've gone for that biopsy, and we've got a different score that rules them out of the study currently, there are a lot more studies now with milder disease that we're looking at more non-invasively. So we've got a cohort of patients with biopsy-proven milder disease, so they may well be eligible for those studies. Biopsy itself is a big deterrent for patients. So whilst they do want to facilitate and participate in clinical studies where they need to, they will also do their utmost to try and improve their own health if that's an option prior to the study. So I think there's a double whammy sometimes of trying to recruit into these studies with biopsy and non-invasive. The more we move to non-invasive markers to monitor and look at outcome generation, then the more we will get a lot more of these patients in and we can look at it from that. But there is a double whammy of trying to recruit and trying to help somebody's healthcare as well.
Yeah, that's an interesting thought. You would think that sticking a harpoon in the side of somebody and telling them that they have disease or that they don't have disease would be a real motivator. Unfortunately, I'm not sure I've seen that pan out in clinical practice. I think the times I've seen it help is when we tell them they have really bad disease. But quite frankly, that could have likely been told to them on a non-invasive test. And as you mentioned, the double whammy could be you go to a biopsy and you tell them they really don't have much disease. Well, then they feel like, well, I guess those potato chips and French fries and tortillas and chips and queso that I'm eating isn't that bad after all. My liver looks okay. Despite the fact that, remember, we're taking 1/50,000th of the liver, but we're only looking at 1 H&E, which is 1/14th of 1/50,000th, which is really 1/700,000th of their liver, and rendering that opinion. So just as we could overstage disease, we can understage it as well. And I think at that point we might be doing them a disservice. But just circling back to the topic of the conversation, that is what has changed, in the past year with clinical trials. I think it is that we're screening more people off NITs than historical biopsies, for sure, either because our practice patterns have changed or the warehousing days are over. We don't have a bunch of biopsied patients just waiting for a clinical trial. But I think more so the mentality has switched away from doing liver biopsy and more to non-invasive tests, and that's fine. But I think the next step is refining which NIT to use in clinical trials based on the population we're studying and which particular cutoffs work, and being malleable enough in the middle of a clinical trial to assess the efficacy of your pre-screening strategy and adjust fire along the way where needed to optimize screen fail rates. But let's be real, I think as long as we have liver biopsy, there's going to be an ongoing issue with heterogeneity, an ongoing issue with interpretation and sampling variability. So there's only so much we can mitigate that screen fail rate with NITs. At some point, the only way we're really going to be able to leave a big fat dent on that is by shifting to a new criteria histopathologically or to an NIT.
The new histopathological criterion is probably for another day in the near future because it's something I think we really do need. If you go back to the idea that goes success breeds success, so the fewer patients you can fail inappropriately, and at the same time, the better job you can do of getting the right population into the trial, that just would seem to breed a whole bunch of success on a whole bunch of levels. And that makes infinite sense. There was one other thing I was hoping we'd get to touch on a little bit today, which Stephen, you kind of started tiptoeing into, and then we pull back and out, let's just dive in, which is if you keep going to the same locations to recruit your patients, there are literally going to be tens of millions of people in the U.S. alone who never get access to anybody who knows anything about clinical trials that are ongoing because they're just not in that loop, right? So if we broaden the loop at the top and don't improve expertise at the top of the screening process, then going back to the psychology problem, you've got results that have low stimulus power. But if you can broaden the funnel at the top at the same time that you're improving your ability to qualify people appropriately for trials, then you can start to massively increase the supply of patients that are available. Now, that's not a 2-week process, but none of this has been a 2-week process. So I'm wondering what steps we're taking to actually broaden the number of patients that get into the top of the funnel. Is there progress being made in those areas right now we're talking about?
Well, I think there's real opportunity here. And look, it's not just about broadening the funnel. It's also about disparities in clinical trial enrollment. I mean, we've gotten a little better at it, but we underrepresent Hispanics, we underrepresent African Americans, other demographics, Samoans. We just assume that the drugs are going to work equally well, despite knowing genetic predispositions are widely varied amongst these different ethnic and tribal groups. So broadening that aperture not only would potentially improve the disparity in trial enrollment across these different racial and ethnic groups, but also increase the throughput. So how do we do that? Well, I propose to have the ideal answer, but one of the areas that we're focusing on in Texas and in Kansas City, quite frankly, to try to mitigate this is something called decentralized trials. This is where we look outside of a referral area for a brick and mortar center, and we go after areas that heretofore have never referred patients to clinical trials because they don't know about them or because the referral center is too far away. And we set up, a central area where we have an office, a small office, and then a couple concentric rings around that small office. We target primary care, endocrine, GI, cardiology, podiatry clinics, and really say, look, we are doing metabolic studies, obesity, diabetes, NASH, and we'd like to educate you about this and about the opportunity for your patients and become a referral center for you where we can actually either go to the patient's house and do the screening, or we can provide a situation where they could come to a closer office area, either through public transportation that's available in the city or through Uber Health or other mechanisms. So we can either go to the patient's house, or if they don't feel comfortable with us doing that, we're at a central location much closer to you, and they could come to us either through public transportation, their own transportation, or something else. And in doing that, you're able to educate a wider population of physicians, and you're able to provide that level of care that heretofore has not been available to them, and you're also able to increase the throughput. One of the peripheral tangential benefits of something like that is when you get a patient that has never been involved in clinical trials, or a provider that has never seen a situation where clinical trials has been helpful, and they have a positive experience, now all of a sudden those providers circle back to other providers and begin to spread that good news. For patients that come in and experience that, they go back to their communities and say, hey, brother, sister, mom, dad, cousin, uncle, aunt, this is what I just went through. You've been telling me you've had the same problem for years. Maybe you need to go see them too. And then vicariously that opportunity spreads. So there's like an organic way that it spreads, and there's like more of a inorganic or peripheral way in which the information about that center spreads. But the problem is there are thousands and thousands of cities in the U.S. and around the world where you could adopt such a practice, but very little people currently doing it. So if you want to talk about where these clinical trials go from here, not just NASH, but any other therapeutic area, this is that great opportunity. The analogy would be you're Lewis and Clark looking out over the vast expanse of the United States, and that's unclaimed territory. You don't know what's over the next hill. And so what comes with that is opportunity and also threat. And what do we not know? What is the risk of that? Are we going to open up a situation where we don't fully know, we don't have a historical basis from which to handle that? So, we can talk about the opportunity, but we also need to be cognizant of the risk and how to mitigate those risks if they arise.
So, Stephen, with an eye on the clock, and then I have a question for Jörn and a question for Louise. Would some of the better tertiary care research universities that have their own primary practices be in a position to do that? I know, for example, a lot of that gets done in oncology. I get all my care in the University of Pennsylvania system. I never got recruited for an oncology trial out of my local primary care office. But, back in the days when I was thinking a lot more about oncology, I kept wondering, why aren't they recruiting for trials out of here? And then I learned, in fact, that some of the lead PIs were starting to move into, in addition to Center City, Philadelphia, one or another of their various satellite offices from where they were recruiting patients. Cancer's an easier target, and a lot more people do the research, but is that something you could see anyone from a Mayo Clinic to the University of Pennsylvania over to a whoever doing?
Yeah, the example that comes to mind, I recently visited Dr. Nagachalasani at Indiana University, and if you look at the state of Indiana, the Indiana University has satellite clinics throughout the state. You could envision a situation where you have a protocol at the main campus that could be fed to all these satellite clinics, and patients could learn about trials that way, and you could have sub-investigators at these satellite clinics that could theoretically do the screening, or they could set up a day and say, look, look, if you're interested in this trial, we're going to have the main guys come down from the main university hub in a van, and they're going to screen you at this particular location on this particular day if you're interested in the trial. And there's all kinds of perturbations of that you could think of. But it starts with what opportunities exist within that ecosystem of the university, whether it's Pennsylvania, the Mayo Clinic, Indiana, and I suspect, Jörn, at your place, very similar type situation.
So, Stephen, you get my $1 for the guy who asks the next question before I get to ask it, or gal. Jörn, how about your place?
It's more difficult, I would think, because the universities are not ready to set up hub centers. But I agree with Stephen in that far that this is unclaimed territory. There's clearly a big opportunity here to expand on care and augment clinical trials and come in and claim this area. So I'm full alignment with that. I think the medical ecosystem in Europe being run a little different. The universities are not quite ready to set up these referral centers, and I think this might go more toward private practices. But in general, it holds truth, and it'll be the same thing. And, even living in a smaller country, people have to work. They're not able to go to clinic, spend time there, and go back to work and miss time of their busy days, because they're being moms, dads, or employees. And I think clearly here, going to the patients is the way forward.
So, Louise, take it to England.
I think it's a fantastic initiative, but I agree with Jörn. I think we're not quite set up for it. It takes an awful lot of time to break down traditional barriers. We commented on the podcast the other week that we have a collaboration call out for the Institute of Research in the UK for liver units to help go to remote locations. But my general consensus from sitting at one of the meetings was we're still thinking within the liver mental framework of it, units working to do it, to collaborate rather than collaborating outside of the traditional. We are moving forward. There are lots of opportunities. We're moving forward in the health service to be carbon neutral. Well, if you go to somebody's local location, you reduce the carbon footprint. So there's lots of ways to do this. But just reflecting on something that Stephen said before about centres being able to recruit. I've recently did a FibroScan catch-up list in 5.5 days of 143 patients. 50% of those patients were referred for NAFLD and NASH. But if we look at the figures, that 31% of those patients that we look at just in the NAFLD and NASH population required an XL probe. Not every centre has an XL probe. Not every place with a fibro, so they're already ruled out. But 8.5 and above, 37% of that NAFLD population made that cutoff, but 17% had full-blown levels of cirrhosis. Now, this is a centre with a FibroScan service that can no longer run it because their team are taking in sick liver patients. So we're not only blocking the funnel, we can't see our own patients. And I am sure that's not every unit in the UK that has a FibroScan. But if you look at the British Liver Trust's details, 74% of the country did not have access to FibroScan and pathways. So therefore, of the 26% that do have access, some of them are so far backlogged. There's a reason we can't get these patients into clinical trial screening. We can't even get them into care. So we do have to re-look at our pathways. And yes, we offer one of the new solutions. But if you don't know about these new solutions, you can't tap into them. Mobile locations aren't a problem for us. Orkneys, Skye, we can get there. We can FibroScan these people. We can get their bloods. We can do all of that with teams. So I think Stephen's absolutely right. We do have to think outside of the boxes that we're currently in. We don't recruit enough low socioeconomic areas. So that's where we see more mortality, more liver disease, more opportunity. But we don't necessarily get to those populations to educate, and they deserve that education. They deserve to be included. We do have to look not only for liver and fatty liver trials, but for any clinical trial. If you get that mobile hub working very well, it has huge potential, and particularly, as Stephen was saying, in the communities that are grossly underserved and under-recognised in targeting these drugs. But the argument would be, whose job is that? Is this pharma? Is this doing education? We know that our biggest populations are obesity, type 2 diabetes, so we have some really big players in that market who also look at NASH and NAFLD opportunities. So it's a big ask, because the fear from healthcare is the more we screen and the more we target even the high populations, the more we will find, and the more expensive that's going to be. So it's multifaceted, but I think we now have to think beyond the traditional, because a lot of these patients don't have abnormal liver tests on blood, but they will will be having other markers, signs, symptoms, metabolic symptoms.
Okay, great. So there's one whole more issue I wanted to get to today that we're not going to get to because we're not only at the bottom of the hour, we're slightly past that. So, Stephen, you got to come back and join us and talk about rethinking the entire question. How do you design clinical trials and sequences versus parallel and all that? Save that thought for a different day, hopefully soon. Just closing question. We each kind of sit in a different place, or we look at the system from a different place. What's the most important thing that's going to happen over the next 6 months or a year from the place that you sit that will make this problem better? And if the answer is, I can't see anything, then be a pessimist and go say that.
I think potentially the UK may take a massive step forward. I think at the end of September, NICE are going to finally decide, having had a provisional turning down, a rethinking, they may well approve FibroScan for primary care. And if they do that, that could unblock the blockages currently going through for people even being screened to get to their secondary care providers and specialists. And I think we can then locate patients. So I would hope that's going to happen because there are patients sitting who are not going to get any care without that.
That would be huge. Jörn, go ahead.
Yeah, from my perspective, I think there is still a lot of work to be done in terms of education. We're slowly making progress in terms of educating other physicians that are seeing these patients and sending them. But the big step is going to be forward if there are partners moving into this field. And I think I'd like to reemphasize that I do see big opportunity here to offer healthcare services through referral systems and building this up. And I think if you have partners, this is going to really benefit drug development. You know, as Stephen said, even outside of the liver disease arena, I think this is something that would make a big difference.
6 months, we'll know a lot about drug development. 6 months, we'll know if Caro's drug worked, Poxel's drug worked, if Altimmune's drug is working in fatty liver. We'll know Madrigal's phase 3 trial results, and we'll know icosabutate's trial results. So you're talking 6 huge sets of data that should be out in the next 6 months. So, and they all don't have to be positive. There is one that does need to be positive. If we want to take the next big step for mankind in the field of fatty liver, we need a drug approved. I think if we can get good news, it's very likely that there will be a drug approval. Then I think the speed of momentum picks up in disease awareness, in patients' interest, in funding to do additional non-invasive testing, to continue to refine our algorithms. It kind of all starts with, I think, that hope of a drug approval. It doesn't have to be approved yet, but I think if we get to the point where we all feel confident that the therapeutic index is such that the drug is likely to be approved, then I think a lot of balls begin to move and are put in motion, and they all have, I think, an effect on how we are going to move forward in this field from a disease awareness perspective, from an NIT perspective, from an endpoint perspective. I think all of that revs up its pace a bit.
So, Stephen, I'm going to use a Stephen that you haven't used today, which is what I used to say to clients at moments like this is if you want to change your rules, find more juice. And the question then becomes, is the juice worth the squeeze? The two specific forms the juice is going to take here are cash and optimism. Optimism because it's going to motivate people to step out of the boundaries they know, and cash because it's going to give them the tools to do that. And what will make the juice worth the squeeze is one particular drug succeeding, and then seeing that it's not the only thing that's ever going to succeed. So I'm dead on with your answer, and I'm happy that I got to use your juice squeeze analogy in support of your answer. I feel good about that.
You could always, if you wanted to be a little off, you could say, is the view worth the climb?
This is more about juice and squeeze, though, because it doesn't need this doesn't need a view. We've had plenty of people throwing views at this, and eventually they don't work. This needs juice. Okay, hey, thank you, everybody. Next week I will not be back, but Louise will be driving the entire boat by herself with the aid of, I believe, Dr. Schattenberg and our new associate producer and the rest of the crew that supports me every day. Stephen, are you going to be in either Dublin or Paris?
I plan to be in Dublin.
Okay. We're doing an episode from Dublin. Maybe we'll find a way to get engaged on site for that, at least for a half hour of it.
I know Jörn will be in Dublin.
Yes, I know Jörn will be in Dublin. I know Jörn will be in Paris, and I'll be in Paris. Louise is not going to be in Dublin. We're working on Paris.
I'm virtual.
Yeah, that's true. And we've all been virtual for a long time. Thanks, everybody. I'll see podcast crew in 2 weeks when we come back, and we will have Stephen back then because we're talking about combination drug therapies with Stephen and Naeem.
By the way, I have to tell Louise this before I forget. I've learned more about your country by watching the show Viking.
Don't go there. My husband's done a DNA test. He is Viking, and Enderby is a Viking name from Danelaw. So
I learned you guys have this holiday called St. Bryce's Day, which I have no idea what that is, but
Neither have I. But being partly Manx, which is a Viking island, and the oldest parliament in the world is Tynwald, continuous over 1,000 years. Viking, you should come from Valhalla. And but they burn a boat for you, so you can come and do Vikings.
Valhalla, I hear, is you have to die to get to Valhalla. So I can go to Uppsala because I hear that's a fun place to go. But Valhalla, I probably will pass on for now. Although I did learn the name Ragnar Lothbrok over and over again. Apparently, he and Leif Erikson are two very important Vikings of yesterday.
Well, Stephen, thanks. I was hunting for my next TV series. I think you just solved it for me. It sounds like a great watch.
No, you would like it. Kid, I'm waiting for a German one. There's got to be some way I can learn about German history as well.
There was an interesting one which was called Babylon Berlin. It was detailing the Berlin years leading up to the Nazi regiment's overtake. A lot of, the Berlin of the 30s was a very interesting time, I think.
Now, I've read a lot about that. I'm a World War II history buff, but I'm talking about taking me back to Roman Empire.
Oh, fighting the Romans? Yeah.
Well, Stephen, you just have to go through because of the Rusks are Viking. So Russians are Viking because it's from the Rusks as they went down through. So, wait till you get through.
I will. Well, thank you guys. Good catching up with you.
Stephen, Jörn, Louise, thanks for a fantastic episode. I think we covered a lot of really interesting territory. I have a lot to think about, and I believe everyone who listens to this will as well. As I say, Louise will be back next week. That's going to be a tremendous experience, and I'll be back with you guys in 2 weeks, along with Stephen again, and Jörn, and Louise, and Mazen, and Naeem, and we're going to be talking about combination therapies. Until then, everybody get a little vacation spirit into your August. If you've not had it yet this summer, stay safe, surf on. We'll be seeing you on the podcast. Bye-bye now.
And now for the Season 3, Episode 41 Business Report.
Slava Ukraini, Heroyam Slava. With all the other news that's been crowding Ukraine off the front pages this last couple of weeks, let's make sure to pay attention to the gallant fight they are making for their country.
Our CME podcast is officially live.
The soft launch for our CME proof of concept, new data, how NITs can help diagnose and manage NASH patients, which I described last week, went fine. So we're now live. This week, we are conducting permission marketing through LinkedIn and Instagram, and we will add paid promotion on LinkedIn and other outlets in the next week. The panel includes Mazen Nuruddin, Alina Allen, Quentin Anstey, and Jörn Schattenberg discussing how best to utilize NITs, sharing data about clinical research and practical advice in equal measures. The way you earn CME credit is by answering thought-provoking questions after the podcast about how to use what you just learned in your day-to-day practice. To access the podcast and eventually the CME, go to the surfingnash.com website, look for the CME link in the upper left-hand corner on the top ribbon, click and follow.
Rising Tide Episode 2 will be out this week.
The next episode in our Rising Tide Diabetes series will be released by Friday. This episode discusses the guidelines that the American Association of Clinical Endocrinology, or ACE, published in May. Our panel includes the lead author on those guidelines, Dr. Ken Kuzi, the third author, Dr. Deanna Barrow, and the two AASLD representatives in that guideline exercise, Drs. Maru Ranella and Zobair Yanasi. They discuss the factors that went into developing these guidelines, the key takeaways from them, and how they suggest these should be used in clinical practice. The target audience is physicians and allied health professionals with large type 2 diabetes or obesity elements in their practices. We will discuss the last two episodes in this series when I return from my vacation in 2 weeks.
Next week, Roger will be on vacation. Louise and the Surfing Nash crew will be in charge.
As we just mentioned, I'm trying something completely new this week, a total week off. Louise will be running the podcast with the panel she described during our episode today, and our new associate producer, Jake Norman, will be thrown into the fire helping her produce materials for the various print and audio outputs, again supported by the magnificent Mike Wilson and Eric Rounds. I'll be back in 2 weeks to discuss combination drug therapies and their likely role in treating patients, along with Stephen Harrison, Naeem Al-Khoury, Mazen Nuruddin, and of course, Louise and Jörn. And with that, I'm off. Thanks to the crew, Magic Mike, Eric, Steve, and Jake, for everything they do and for the peace of mind I anticipate. And as always, thanks to all of you for making this such a worthwhile labor to pursue. Remember to look on the Surfing Nash website for the links to the CME pilot today and the new Rising Tide and Diabetes episode later in the week. Stay safe, surf on. I look forward to seeing you in 2 weeks on the podcast. Bye-bye now.
Have any questions for the surfers? You can send them to surfingnash.com, and we will answer on the podcast or the website.